Hey there, future doctors. Are you ready? Let's talk about biological classification. थोड़ा डर लगता है इस चैप्टर से? It's okay. चलो, आज इसका एकदम rapid fire revision करते हैं, focusing only on the high yield questions जो सीधे NEET में आते हैं. Let's dive in. I know, biological classification में थोड़ा confusion होता है ना? इतने सारे names, इतना सारा data, it can get overwhelming. But hey, don't you worry. In the next few minutes, we are going to crack the most important questions from this chapter together. By the end, you feel way more confident. Ready to do this? Okay, so Section 1, Classification, Kyun or Kaise. Let's go all the way back to the beginning and see how this whole thing even started. We're talking about the very first attempts. Right, first question. Think about it for a second. Who was the very first person to try and group living things, not just for, food or shelter, but on an actual scientific basis? Any guesses? The answer is Aristotle. That's right. He used simple morphological characters, which is just a fancy way of saying he classified them based on how they looked from the outside. Super simple. He divided plants by their size into trees, shrubs, and herbs. And for animals, he just made two groups, those with red blood and those without. It might seem basic now, but back then, it was a revolutionary start. So after Aristotle, the next major step forward was the two-kingdom classification. The question for you is, who came up with this system? And more importantly, what were its flaws? What was so wrong with it that we had to move on? This system was given by Linnaeus. He just made two huge kingdoms, Plantae and Animalia. That's it. It was simple, sure, but the problems were massive. I mean, it mixed up prokaryotes and eukaryotes. It threw unicellular and multicellular organisms together. And the biggest mistake of all, it put photosynthetic algae and non-photosynthetic fungi in the same group. Clearly, this system was just way too simple for the incredible diversity of life on Earth. And to solve all of these problems, a hero enters the story. R.H. Whittaker. His five-kingdom classification is the absolute gold standard, the one we still follow for NEET. So pay close attention, because this is the most important part of the chapter. So here's the next big question for you. What exactly did Whittaker do differently? What were the main criteria he used for his classification? Because, these criteria are the real strength of his entire system. And here they are. Just look at these 5 points. They solve every single problem from Linnaeus' system. Whittaker looked at cell structure, body organization, mode of nutrition, reproduction, and the big one, phylogenetic relationships, which is basically their evolutionary history. Based on these 5 things, everything just fell perfectly into place. And this is the amazing result, the ultimate comparison table. Okay, guys, this table is super important. You get direct questions from this. Seriously, take a screenshot of this right now. Just look. Monera is prokaryotic. Everyone else is eukaryotic. Fungi have a cell wall made of chitin, while plants have cellulose. These are the exact details they ask about in NEET. Okay, Section 3, Monera and Protista. Let's put on our virtual lab coats and take a deep dive into these incredible microscopic worlds. So, Kingdom Monera, its sole members are bacteria, and you get direct questions on their shapes all the time. So, what are the 4 basic shapes of bacteria? Try to recall them. Boom! Here they are. Coccus, which means spherical or round, bacillus, which is rod-shaped, vibrio, which is comma-shaped, and spirillum, which is, spiral-shaped. Simple, right? You just have to remember these 4 names. Now, in the Monera kingdom, there's another superstar, mycoplasma. This one is an exam favorite. Why? Because these are the smallest living cells known, they have absolutely no cell wall, and they can even survive without oxygen. They're also called the jokers of the plant kingdom, because without a cell wall, they don't have a fixed shape. After Monera, we get to Protista, which includes all the single-celled eukaryotes. You can kind of think of this kingdom as a bridge, It connects Monera with the other more advanced kingdoms. So what are its major groups? Here are the 5 major groups of Protista. Let's quickly run through the most important must-know points about each one. Check out this diversity. Chrysophytes have indestructible silica walls which form diatomaceous earth. Dinoflagellates are the ones that cause those infamous red tides. Euglenoids are mixotrophic, autotrophs in the light, heterotrophs in the dark, and protozoans are basically the primitive relatives of animals. These are your key matching points right here. And now, Section 4, Kingdom Fungi. Let's talk about the great decomposers, the recyclers of our planet. The sexual cycle in fungi has 3 steps, and the sequence is extremely important. You have to remember the order. First, plasmogamy, which is the fusion of protoplasm, then karyogamy, the fusion of the nuclei, and finally, meiosis in the zygote, which forms haploid spores. Just remember PKM, plasmogamy, karyogamy, meiosis. Got it? So, with so many types of fungi, how do we classify them? Well, it's based on these 3 things: the morphology of the mycelium, the mode of spore formation, and the fruiting bodies. These 3 criteria are what give us the 4 classes of fungi. Okay, our 5th and final section, viruses, viroids, and prions. Now we're talking about the misfits, the ones that Whittaker didn't even give a spot in his kingdoms because they're acellular. This is a classic question, and honestly, a place where a lot of students get confused. What is the fundamental difference between these 3 infectious agents? What are they actually made of? This table will clear up all your confusion. Just look. A virus is a protein coat plus genetic material. A viroid is just free RNA with no protein coat at all. And a prion, it's just an abnormally folded protein. That's it. Remember, viroids were discovered by T.O. Diner, and prions cause those scary neurological diseases. This is a must-know slide. And we'll end with this final mind-bending thought. Viruses are basically non-living crystals outside of a host, but once they get inside, they replicate just like a living thing. So here's the question: can we actually call them living? This question really challenges the very definition of life itself. Think about it. All the very best for your preparation.